Good morning, children. In the previous class, we have seen about the different parts of the microscope. About the scientist, we have learned everything regarding the cell, what is the basic and structural unit of life, as it is the cell you have known it. Is it clear, children? If you see this on the board, the picture, I think you can easily identify that it's a Robert Hooke's microscope. It's a Robert Hooke microscope. From this, you identify the parts. This is the eyepiece. Is it clear, children? Through which the object is being observed. This is the eyepiece. Next, coming to the barrel, the cylindrical shape that is present here. This is the barrel, and this is the fine focus screw. To make the object visible clearly, you will adjust the fine focus and coarse focus screws where you find in his microscope, Robert Hooke's microscope, fine focus screw. And this is an objective through which the object is being observed. This is an objective. And next, coming to this here, this is the specimen holder. The specimen holder itself tells you that the specimen, that is the object, is kept over this holder. That is the place where the object is placed. It is called as the specimen holder. Okay, it's stage that you have learned in the previous class, isn't it? So that is also called as a specimen holder. And this is an oil lamp, and it is used as a source of light. And here the water flask, which reflects the object through the source of the light. So these are the parts of the Robert Hooke's microscope. Come on, children. What is this? It's a Robert Hooke's microscope. Next, coming to the parts of the plant and animal cells. That is, there are two different classifications of the living organisms as plants and animals. If you identify them and observe the cells of each under the microscope, you can differentiate between each of them. How to do it? For this, we have to observe the plant cells or the animal cells under the microscope. As a laboratory experiment, we can do this by taking an onion peel or a dead cell of matchstick. And also we can see that it's a dead cell by seeing this under the microscope. How to do it? I'll explain you now. Okay, children, to know the dead cells. This is a matchstick. Okay, children, so this is a matchstick. If you soak it in a water, it becomes easier for us to peel its stem. So, if it is soaked, you can easily peel this. I have already told you about the different, that is the preparation of the slide, how to use the object over the slide, glass slide, isn't it? So, you have to cut it with a sharp blade or razor in such a manner that 0.5 thickness or can be cut easily. Is it clear, children? See here, it can be cut. The stem is being cut. And if you put this under the microscope, is it clear, children? Under the microscope, you can easily observe with this. So, very thin slide object to be taken, to be observed under the microscope to see its cells. Okay, children, if you observe this, if you keep this object over the stage or the specimen holder and observe through the objective lens from the eyepiece, you can observe the cell parts of this matchstick. Matchstick you get from the tree that is dead, isn't clear? So, we can observe the dead cells of the matchstick in this manner, children. rectangular shaped cells that you observe in the dead cells of the matchstick. Did you understand, ma? These are the dead cells. Okay, children. Generally, this is being observed in by this is very similar to that of the cork cells that the Robert Hooke has observed under his microscope. So he said it's a honeycomb structure that he observed is like a honeycomb structure, like cavities, that is a cells. He called it as a cavity, like a room, little room it is, isn't it? So you cannot find any nucleus in it. It's completely with the cytoplasm inside. Is it clear, children? So this is of the Robert Hooke's observation under the cork cells. This is an observation of the matchstick which showed under the microscope, it showed the rectangular shaped cells. You can see the rectangular shaped cells under the microscope. Is it clear? So this is about the dead cells. Then what about the plant cells, or if you observe the onion? So if you take an onion and peel it, so you can say how to peel the onion. So if you observe an onion and take a tiny part apart from the onion and peel it with a sharp blade or any knife, anything, you can peel it, a thin layer of the onion if you take it and keep it over this slide, rectangular glass slide, and cover it, keep over the glass, keep the object, that is the onion peel. You have taken a very thin specimen of it and you have kept on the glass slide and you have covered it with the cover slip. Before that, you have to add a drop of water over it to make it not to dry for if it is kept for a longer period. I have already told you about the precautions that you have to take while you are preparing a slide, isn't it? So after taking the onion peel over the glass slide, a tiny, that is the thin film of the onion you have to take and keep it over the glass slide by a drop of water on it, not to get dried. Then cover it with a cover slip. If you want to see the cells, that is the differentiate the different types of cells are in it, you can add a little bit of, already it's a pink in color, you can easily observe it. If it is any other item, if you are going to observe it, if it is in transparent, you can add the saffronin or red ink or any other indicators to observe the tissues or any other parts inside the cell. So you can observe how the onion peels will be observed is in the form, you can easily see, I'll show you here. They are in this manner. This is a plant cell. Onion is a plant cell. It has a cell wall. It's a and there is a nucleus in every room, that is, you can say that it contains the cell in the peel. Inside the peel, the cell that is it is represented by a cell. As Robert Hooke said that the rectangular shaped parts that we see are in the form of a little rooms, isn't it? So these are this is cell wall, parts of the onion peel cell. Okay, onion peel cells. If you observe under the microscope, how to observe I am telling you. Onion peels, it contains the nucleus, cytoplasm. So this is a nucleus. You can see at the periphery or edges, you can see a nucleus in every cell. Nucleus is present in the periphery, and the cytoplasm. These are the parts of the cell, plant cell. Okay, this is cytoplasm. This is nucleus. This is a cell wall, and this is a cell of the peel. Cell of the peel that you have taken. Is it clear, children? So this is about the onion peel cell. You can see that there is a cell wall. Inside it there is a cell membrane. It's covered by the cell wall. What is the function of cell wall? In every plant there is a cell wall, that is it protects the plant. It protects the plant and gives it rigidity. The cell rigidity, or it gives protection for the plants. And also the cells act as a pressure vessels. That is, the cell walls of the plant cells act as a pressure vessel. What do you mean by it? The pressure vessel, that means when extra water enters into the cell, it helps to mobilize itself. Okay, it not entering the extra water into it and making it enlarge. It helps to, the cell wall helps as a pressure vessel not to enter the extra water into cell and make it expand. Okay, children, and next coming to the animal cells. To observe the animal cells, you need to take that is the chick cells you have to take. How to take this chick cells? You should not rupture completely inside the chick cells. You have to take a blunt spoon. It should not be very sharp. It should be very blunt. The edges should be very blunt. You should scrape into the mouth, from the mouth of your chick, and you have to drag it downwards and take along with the water you get it. You have to put it over the glass slide. I have shown you before, isn't it? As we have kept the onion peel over the glass slide, you will be taking the chick cells also on a glass slide. As usual, you will be observing it under the microscope. So for this, you observe the nucleus. You observe the nucleus in the center and also the cytoplasm. Nucleus in the center and also the cytoplasm. Okay, children, now see here, this is a structure of animal cell, that is the human chick cells. Okay, you can see the cell membrane only in the animal cell, but not the cell wall. So what is the difference between the cell wall and cell membrane? How does the cell wall protects the plant or helps the plant, and why there is no cell wall in the animal? That is the doubt that everyone gets into their mind. For that, you have to know that first, here the cell wall, the shape of the cell, it contains the nucleus, cytoplasm, but only cell membrane, but not cell wall in the animal cell. Okay children, so why is it so? In general, we can say that the plant cell have the cell wall around them. It helps to protect the plant against the different conditions, that is to, and helps to growth of the plant. The cell wall helps for the growth of the plant and to get the sunlight for the preparation of the food. It also gives the shape to the that is the cell. The cell wall, only the plant gets the shape with the shape of the cell wall in itself. So we can say that the plants have cell walls. That is the main difference between the animal cell and the plant cell. We can say the plant cell and animal cell. What is the main difference between the plant cell and animal cell? Here, the plant cell has cell wall. The animal cells has only cell membrane but not cell wall. Only cell membrane but not cell wall. Why? Here, the animal cells, different cells are present in the animals to perform different activities, and the different cells have different shapes. Here, all the cells of the have cell membranes in the animal cells, and they are flexible, that is they are capable of movement. But here in the plants, they cannot move, they are non-motile, isn't it? So the cell wall helps the plant to be rigid and protects it against the external water that enters into it. I already told you that it helps to for the growth of the plant and preparation of the food through the process of photosynthesis by taking in the sunlight. So we can see that it contains the cell wall, and it contains the cell membrane, and it gives, cell wall gives shape and size. Shape and size to the cell. Okay children, and here all the cells of the animal have different shapes. All cells have different shapes. Okay children, animals have various shapes. So the cell wall gives protection against the external pressures, that is it acts as a pressure vessel by mobilizing it to extra water to enter into the cell. So it protects the cell, for protection you can say. And here the cell membrane protects. The cell membrane protects the animal cell, and animal protects itself. It moves around. It moves around and gets its food. So the cell membrane, here there is no need of cell wall. It has the mobility to move around. It has mobility to move and get food. Whereas it is rigid. The rigidity helps for the protection of the cell. Is it clear children? Yes. So we can say from this children that the plant cell differ from the animal cell in having a cell wall in them. So here this is about the plant cell and animal cell. Today we have seen how the differences between the plant cells and animal cells, dead cells. So observation of the onion peel cell, human seed cheek cell, and differences between the plant cell and animal cells. Also we have seen the dead cells of the matchstick. Is it clear, children? So today's topic, you refer once again from the textbook, read it out and make a notes of it. Okay children? Bye children. Have a good day.